Hi, my name is Paul Hatzinger. I'm the director of the Biotechnology Development and Applications Group at Aptum. This presentation today will cover co-metabolism. We'll go through some fundamentals. We'll talk about applications for a variety of different contaminants that are found in groundwater. And then during the second half, we'll actually walk you through a system that's operational, sort of show you how that system was designed and how it functions at field scale. And this project is supported by the ESTCP program under project ER20 5036. Please enjoy.
All right, so let's start with some fundamentals. So what is co-metabolism? Well, this process is the transformation of an organic compound by a microorganism that can't use that substrate as a source of energy or one of its constituent elements. So if we look at this diagram on the left, so this would be a bacterium, and it's degrading a contaminant of concern like benzene aerobically. So that benzene will be degraded by a series of enzyme systems, create carbon dioxide, and the cells will grow, creating new biomass, and they use oxygen as a terminal electron acceptor. So by this process, the cells gain energy and carbon from the contaminant itself. Now, when we look at cometabolic processes, there's a primary substrate that's not your contaminant of concern, and this can often be small ring compounds, things like toluene or benzene, or alkane gases like methane, ethane, and propane. So the cells grow on these primary substrates, form biomass and CO2, but these substrates induce enzymes, and they're often oxygenases that have very broad specificity. So these oxygenases also break down contaminants of concern. So, things like 1,4-dioxane can be broken down by these types of oxygenases. This is again an aerobic process, but in this case, the cells gain the energy from the primary substrate and they use the contaminant of concern fortuitously and don't gain any carbon or nitrogen or energy from that process. Okay, so what compounds are subject to cometabolic biodegradation? And it turns out there's quite a few. Some of the compounds of interest today are things like 1,4-dioxane. Many different pesticides can be broken down cometabolically. And if we look back, this is a 1972 review article on cometabolic degradation, and it cites research that goes back to the late 50s, where scientists noticed that methanotrophs, so bacteria that normally grow on methane, can actually also biodegrade ethane, propane, and butane, although they don't get any energy from this process. So these were some early studies on co-metabolism. But among these early studies, dating to the late 1960s, you start to see research on some pesticidal compounds and and this research shows that those compounds also were co-metabolized. And there was a lot of work that went on, in the 70s and 80s, just showing, the cometabolic biodegradation of different pesticide compounds. But you also have things like NDMA or N-nitrosodimethylamine, MTBE, the gasoline oxygenate, many different chlorinated solvents can be degraded this way, and a number of other compounds. And today I'm going to, provide some examples of cometabolic degradation of everything in red here, 1,4-dioxane, a couple of different pesticides, and then NDMA and some chlorinated solvents. So there's a very wide variety of cometabolic enzymes and a tremendous diversity, and this is likely the tip of the iceberg. Scientists are finding more and more of these types of enzymes. But some that you see on the list are soluble in particulate methane monooxygenase. These are enzymes possessed by methanotrophic bacteria, those that grow on methane. Ammonia monooxygenase, some different types of oxygenases that break down toluene and propane. Butane, some different alkene monooxygenases, and then a range of others as we move along that all have these types of cometabolic activities. So historically, it was thought that these organisms may have had one or possibly two oxygenases, but research over the past several years has shown that some of these different types of bacteria have many different oxygenases, and that more than one can be induced and active at the same time. So this strain, for instance, strain NBB4, this is some work by Nick Coleman's group, has at least six distinct monooxygenases. And it will grow on things like C2 to C4 alkenes, C2 to C16 alkanes, and it degrades a whole variety of different compounds, including 1,4-dioxane and some different chlorinated solvents. And here's an example of this organism that was some work we did in our lab. And you see this organism was grown up on ethene, and it biodegrades cis-DCE or cis-dichloroethene very quickly. But in the presence of ethene gas, it doesn't degrade. And often this is what you see. If you have a high level of the primary substrate, in this case ethene, you get a competitive inhibition, so you don't see that contaminant of concern degraded until the ethene itself is biodegraded. Interestingly, this strain also degrades 1,4-dioxane, but the pattern looks a little different. So in this case, we see the 1,4-dioxane being degraded in the presence or the absence of ethene. And what that suggests is this may be two different enzymes. So it's possible that when you feed this organism ethene, it induces multiple enzymes, and it's growing on ethene, but a second enzyme is also induced that's degrading 1,4-dioxane. And we're actually doing a little work now to try and understand this a bit better. Okay, then, so what are some of the advantages of co-metabolism, or, when can it be best applied? In my mind, one of the best applications of this approach is when you have very dilute contaminants or dilute plumes of different types of constituents. With growth-linked metabolism, when you have dilute contaminants and the organisms have to grow on them, it's very hard to induce cell growth and increase biomass. And as an example of this, in the United States, more than 90% of Air Force sites have less than 120 ppb of 1,4-dioxane. So it's very hard, even though there are organisms that can grow on 1,4-dioxane, it's very, very hard to grow them when concentrations are so low. Now with co-metabolism, growth is abundant on a primary substrate, so you're actually adding a substrate, let's say, to an aquifer, so you can really get high biomass that way. And there are times when you can achieve very low contaminant concentrations. And this is a nice example of that with the molecule NDMA, or N-nitrosodimethylamine. And this is a culture isolated in our lab called Rhodococcus ruber ENV425. It's bright orange. And you can see in this flash study, this organism degrades NDMA from 8,000 nanograms per liter, or 8 ppb, down to less than 2 nanograms per liter. So it achieves very low concentrations. These organisms are also very widely distributed in the environment. So I'm going to move into some case studies shortly, but before I do that, I just want to talk about some important considerations with cometabolic processes. Now, typically, when you're applying co-metabolism in the field, what you're doing is you're adding a carbon source, often a gaseous compound, something like propane or methane or ethane, to groundwater. And in doing so, you're adding quite a bit of carbon. So you always have to consider whether there's enough inorganic nutrients, things like nitrogen and phosphorus. And here's an example of a microcosm study. And in this case, we were doing some work looking at substrates that support the biodegradation of 1,2-dibromoethane, EDB. I'll talk about this a little further in a case study. But it's a soil fumigant. It was also used in leaded fuels. But in this microcosm study, if we just added ethane or propane, we saw no biodegradation of EDB, and this is at 60 parts per billion in these bottles. But if we added a nutrient source like diammonium phosphate, we saw good biodegradation of the EDB. In fact, moving a little further, we actually saw concentrations below 15 nanograms per liter. So very, very good degradation of the compound. We didn't see that if we just added the nutrients. So clearly there was a nutrient limitation that had to be overcome. So the next important consideration is, which substrate should I use? And there are a variety of different substrates that can be applied to stimulate co-metabolic processes, and they can stimulate different organisms, or there are cases where perhaps the organisms aren't in the aquifer that you're looking for. So maybe you don't have propane degraders, but you do have good populations of methanotrophs. So it's very good to run a microcosm study, take a look at which substrates are most effective, what are the relative rates. And if you have multi-contaminants, sometimes one substrate will allow you to biodegrade all of those contaminants. And there are cases where you can use multiple substrates. So you could use methane, for instance, to stimulate the degradation of TCE, and then ethane for 1,4-dioxane or something like that. So you can sometimes apply multiple substrates. The other thing that's important is actually the properties of the gas itself. Its aqueous solubility, its lower explosive level, vapor pressure, and purity, as I'll discuss in a second. But some gases, you want gases typically that have a good vapor pressure so they volatilize easily and can be sort of readily added to an aquifer in the gas or vapor phase. So here's a nice example of different organisms degrading different contaminants of concern. So this is an organism that was isolated on isobutane in our laboratory. It grows beautifully on isobutane, and when doing so, it degrades 1,4-dioxane very readily. But the organism won't degrade trichloroethylene at the same time if you grow it on isobutane. So it degrades 1,4-D, but not TCE. Now, if we look at this other organism, it's a well-known methanotroph, so it grows on methane and it has soluble and particulate methane monooxygenase. This organism does not degrade 1,4-dioxane, and most methanotrophs don't, but it does degrade TCE. So an example where you have two different contaminants and you get very different results depending on which gas you use or which organism that you're looking at. Another important consideration is gas purity, and it's often best if you're conducting a project using co-metabolism to use relatively pure gases, usually at least 99%. And this is just an example. This is a typical grade of propane called LP gas. It has a lot of propylene, which is not very good for growth of some of these organisms. It also has mercaptans, which they don't like. And this is just an example of one of the organisms that we work with growing on pure propane. So this is optical density, so increase in density of cells versus consumer grade or LP propane. And it just doesn't grow at all on the LP propane where it grows beautifully on pure propane gas. Okay, so with cometabolic processes, you're often trying to introduce gases into an aquifer to stimulate growth of bacteria. So how can you do that? There's a couple of different ways. You can use what's called active biosparging. So you blend a low concentration of your gas into air, usually with a compressor. You inject that into the ground in pulses. It's very flexible. You don't have to pump any water. You usually have a good radius of influence. You can use that in confined and unconfined aquifers, although sometimes you'll have some preferential flow paths, which is a negative for that. You can do a recirculation. So you pump groundwater from the ground, add your nutrients and your co-metabolic substrate, and then repump it into an injection well. So you create a bioactive zone between these. Very flexible. You can get good amendment distribution. You can also add cultures and liquid nutrients. You have to watch out for biofouling, and of course you need to have a pumping infrastructure to apply this. And the third approach is passive in-well diffusion, where you can actually put types of tubing. There's something called a Waterloo emitter that you could put into wells and just put gas pressure on these. This is a very simple design, but typically the radius of influence is pretty poor. There are cases where this can be applied, but often the radius of influence is so small that it's not particularly effective over a reasonably large area.
So now we're going to move into some case studies. I'm going to talk a little bit about in situ treatment of EDB using groundwater recirculation. I'm going to go through a study where we treated 1,4-dioxane and dilute CVOCs using propane biosparging. Just touch a little bit on another biosparging system for CVOCs. And then we're going to walk you through a hybrid solar powered system that employs both groundwater recirculation and pumping, as well as two different gaseous substrates. So we actually took some video on site. So myself and Dave Lippincott will actually kind of walk you through how that system is designed and show you how that system operates.
Okay, so the first case study involves 1,2-dibromoethane, or EDB. Now, this compound was widely used in the US as a fumigant for nematodes, and it was also a lead scavenger in leaded fuels, including aviation gas. It's a potent carcinogen. It has a federal drinking water standard of 50 parts per trillion, or only 0.05 ppb. It was phased out in the late 1980s for both uses in the United States, so both as a fumigant and a lead scavenger when it was found to be a very potent carcinogen. But it persists at many thousands of gasoline sites in particular at concentrations above this MCL, this 50 part per trillion MCL. It also persists at some former sites where it was used as a fumigant. Characteristically, it has a very high solubility, it has a low KOW, so octanol water partitioning coefficient, low Henry's law. So what that means is it's hard to remove from water. It's hard to strip. It's hard to adsorb to carbon. Microbially, there are some organisms that grow on it, but concentrations are very, very low in groundwater. So again, as I mentioned earlier, it'd be atypical to have a site where this contaminant is at such high concentrations that bacteria will grow on it. But there are a number of substrates that can promote co-metabolism, including things like propane, ethane, methane, and ammonium. So this was a field pilot recirculation system for EDB. And basically there was a plume, it was a former leaded fuel spill, and all of the hydrocarbons had been cleaned up, but there was EDB that had moved down gradient from this spill. So we did an in situ pilot, and through some microcosm studies, and I showed you some of those earlier, we found that ethane gas promoted natural bacteria to break down EDB. So we designed a system where we pumped water from an existing extraction well at about 38 liters per minute. We amended it with ethane, oxygen, and diammonium phosphate as a nutrient, and then re-injected it into the ground. So we created a subsurface bioactive zone in the area where there was EDB. And there was a series of monitoring wells installed between. So the recirculation system was run in different phases. Initially, for about 82 days, we recirculated just to make sure that the EDB was at equilibrium within the aquifer. We then started adding ethane and oxygen. And then around day 120, we started some nutrient addition and then shut the system down after about 209 days. And what we see here is that if we started adding our ethane and oxygen, we see an initial sort of concentration of ethane that reaches about a little under 1.4 ppm at the most concentrated well, so not very high concentrations of gas. A lot of the other wells never got more than about 500 or 600 ppb. But as we run the system and bacteria start to consume this gas, you see by the time we hit 4 or 5 months of operation, there's very little detectable ethane in the system because they're consuming it all as it moves through the aquifer. Now, if we look at the field data for EDB, we can see in this aquifer there was only 300 parts per trillion, or 0.3 ppb. As we did the preliminary mixing, we see that everything's sort of pretty much grouped in that area. Then we start our ethane and oxygen addition, our nutrients, and we see very good declines in EDB in all of the well-connected monitoring wells. This was actually the water that was coming through the plot, so all of the water that was coming out of the extraction well, and we saw a general decline there as well, which was quite interesting. When we shut the system down, we see some rebound in some of the wells, but it takes a fairly long time, and there seems to be continued activity in some of these wells over, a period of almost another 100 days. At day 209, when we shut the system down, you can see these were the concentrations in all of the well-connected wells, and we had achieved what was the Massachusetts MCL of only 20 parts per trillion in all of these different wells. And we also isolated a bacterium that grows on ethane and degrades EDB, and that we're studying in our laboratory right now. I just wanted to slip in another quick example after the dibromoethane of 1,2,3-trichloropropane, which was also used as a soil fumigant, kind of similar to EDB, and it also can be co-metabolized by a variety of different bacteria. This was an organism, again, that we had isolated from an aquifer in California, and when this organism grows on isobutane, it degrades 1,2,3-trichloropropane. It'll also grow on propane gas, and you see it doesn't degrade it quite as quickly. And you'll see a little degradation on isobutanol, so probably one of the enzymes that degrades trichloropropane, so one of the oxygenases, and we're not sure which one it is at this point, is induced a little bit when the organism's growing on isobutanol. The other example is 1,2-dichloropropane, which we were studying, which the organism degraded very readily, irrespective of which substrate it grew on, which was quite interesting. And I'll also mention that this organism was able to degrade trichloropropane from 20 ppb, which is a relevant concentration in some aquifers, to less than 5 nanograms per liter. So my next brief example is a field biosparging system for treating 1,4-dioxane and dilute chlorinated solvents, or CVOCs. 1,4-dioxane is a stabilizer in solvents, including 1,1,1-trichloroethane in particular. It's a likely carcinogen, and it's got a lot of characteristics like EDB. It's soluble in water, very soluble in water. It has a low Henry's law. So it's hard to strip, and it's hard to adsorb to carbon. There are organisms that grow on 1,4-dioxane, and one that's well known is Pseudonocardia dioxinvorin CB1190. But again, concentration in aquifers tend to be so low that it's very hard to apply effectively growth-linked processes. It's been shown to be co-metabolized with propane, ethane, toluene, or isobutane. Now, the case study I'm going to show you was a U.S. Air Force Base site. It was a tank maintenance facility. It has 1,4-dioxane at reasonably high concentrations, about 100 to 1,000 ppb, as well as some low levels of CVOCs. And the goal of this study was to apply in situ biosparging and to treat 1,4-D, and to see if the other co-contaminants could be treated as well, because all of these can be treated co-metabolically. This just shows a little bit of the geology at this site. There are several water-bearing zones. The zone that the demonstration was performed in is known as the deep water-bearing zone. It's well-graded sand with gravel. And one biosparge well was installed, along with a series of monitoring wells, a couple that were short screen wells, and then a slightly longer screened well. And this well was pre-existing and maybe screened across some different intervals. And a well that was up in one of the other intermediate shallow zones was used as a control. So treatability studies that I'm not going to show here due to time showed that propane was an effective substrate here, but propanotroph populations were low, so the aquifer was actually bioaugmented with a propanotroph to try and increase the density of the organisms at the beginning. During the first month, there was only air sparging just to see if there was any stripping of 1,4-dioxane. That occurred at 145-minute pulse per day. And then the propane addition began around day 29, and the aquifer was bioaugmented on day 42. And then from there on out, it was just propane and nutrient addition. Again, very, very low amounts of propane, about less than a pound a day. And basically, after the system was optimized and you see a lot of propane consumption, it ends up being about 6 cycles per day. So the propane is sparged in and then allowed to biodegrade, sparged in, allowed to biodegrade. And there were periodic nutrient additions right into the sparge well of diammonium phosphate. You can also use ammonia gas actually for sparging. So here are the 1,4-dioxane data. This is background concentrations, which ran from about 100 to a little under 1,000 ppb. This is the period where there was just air sparging. You see no consistent losses. And then propane addition starts along with bioaugmentation. And you see very good declines in concentrations in all of the really good connected or well-connected monitoring wells that saw propane. This is 5A, the control well, which is up here. And this 5B well is the broadly screened well. And basically not a lot of propane got to this well. It was probably pulling a lot of water from an upper part of the formation, and there may have actually been sort of a confining layer here. But all of the wells that were well connected to the sparge well showed very good declines in 1,4-dioxane. And you can see what those numbers were. So greater than 99% in 3 wells, 95% in the other, down, basically the concentrations were achieved down into the low ppb range. So this was very successful for treatment of 1,4-dioxane. As I mentioned, there were also chlorinated solvents at this site, including 1,11-TCA, 1,1-DCE, and TCE. The concentrations overall were relatively low. We see TCE sort of up to the 15 ppb range. For 1,1-DCE, at least down in the zone where the propane was added, we're in the range of 10 to probably a little over 100 ppb, and kind of for TCA, we were mostly less than 10 ppb. Now, the 1,1-DCE story here becomes important, as I'll show you later in kind of the final extensive example of treatment with a hybrid system, because this compound can be relatively toxic. Now, in the case of this site, the concentrations in groundwater, again, in the zone were relatively low, and we do see evidence that it biodegrades in this region. We also see that for both TCE and 1,1-TCA. I should note that samples were taken back to the laboratory just to show that this wasn't sort of some type of sparging effect. And in fact, when nutrients and propane were added in the lab, all of these contaminants biodegraded.
I'm just going to briefly go through a third example. This is an ESTCP project that was funded basically to look at cometabolic treatment of dilute cis-DCE and vinyl chloride. A system was designed which is solar powered, so it's completely off the grid. And the system essentially was made to add gases, in this case propane, oxygen, and ammonia, with the ammonia being a nitrogen source, to a 200-foot barrier cross gradient to groundwater flow. Now the system, as we see on the right, there was a series of operational phases, but most importantly, this barrier system successfully treated both vinyl chloride and cis-DCE to below relevant MCLs. And if you'd like to see more information, there's a project report on the CertUp ESTCP website under ER 201629.
So the last example today is going to be rather extensive, including some videos, and this goes over the design and operation of a hybrid biosparging groundwater recirculation cometabolic system. This system was designed to treat a mixture of chlorinated solvents and 1,4-dioxane at a site, and it actually employed multiple substrates, so multiple cometabolic substrates, in this case isobutane and methane. The system was actually a retrofit of the previous system that I showed you. So we took the off-the-grid biosparging system that I showed you in the previous example, and then basically retrofit that system so it could also recirculate groundwater. This project was funded by ESTCP and led by Tony Danko at NAVFAC XWIC. Also included myself, Dave Lippincott, and Greg Livornia from Aptum, Lou Semprini from OSU, and Mike Hyman from North Carolina State. So the demonstration site where this project was conducted had a complex mix of contaminants, including 1,4-dioxane and an array of CVOCs. Unlike the Air Force site that I talked about previously, the chlorinated solvents at this site were present at a higher concentration. In particular, TCE and 1,1-DCE were there, at many hundreds of ppb compared to kind of single-digit ppb, maybe to 100 at the Air Force site. Now, of particular interest here is 1,1-DCE because, as I mentioned previously, this compound can form a relatively reactive epoxide when it's biodegraded by cometabolic organisms. That epoxide then can react with cellular materials and actually kill the cells that are degrading it. So in this case, we sent some aquifer materials to Lou Semprini, who set up microcosms at Oregon State. They added isobutane as a cometabolic substrate along with inorganic nutrients and oxygen, and basically saw no biodegradation for a period of about 3 months. Now, simultaneously at our lab in New Jersey, we were doing some isolation work, and basically we enriched a culture, which we then purified, which was a Rhodococcus strain called ENV493. And when grown on isobutane, that organism could degrade this entire suite of contaminants with the exception of TCE, which it did not degrade terribly readily. So we sent a sample of this culture to Lou Semprini's lab at Oregon State. They bioaugmented the microcosms, and lo and behold, cis-DCE degraded 1,1-DCE degraded very, very quickly. So when enough of this culture was added to the samples, it degraded 1,1-DCE to non-detect. Now, the other contaminants of concern, including chloroform, 1,4-dioxane, and cis-DCE, were also degraded by 493. And in particular, when the 1,1-DCE was gone, the strain then degraded all of these other contaminants of concern. The one exception was TCE, and ENV 493 degrades that relatively slowly, but we actually enriched a consortia of methanotrophic bacteria, so those are organisms that grow on methane from the site, that could degrade TCE very, very readily. So now we had sort of an interesting scenario where we had two different organisms that, when bioaugmented into site samples, growing on different substrates, could actually degrade the entire suite of contaminants at the concentrations that were found in the aquifer. So we used this information as we moved into the field demonstration phase, as I'll talk about in the next slide.
So based upon our laboratory findings, we basically understood that if we just added gases to this aquifer, isobutane, methane, any other cometabolic gases, we were unlikely to see biodegradation of the contaminants of concern. And we felt the problem here was the fact that 1,1-DCE was causing toxicity to the organisms as they tried to grow. The other thing we understood is that if we had bioaugmentation, so if we could get high enough concentrations of bioaugmentation cultures in the ground, we could potentially overcome this limitation. And in particular, we knew that ENV 493, the isobutane degrader, could actually biodegrade most of the contaminants that were there, including 1,4-D and chloroform and suite of chlorinated ethenes and ethanes, but the organism didn't degrade TCE particularly quickly. However, the methanotrophic culture that we isolated could degrade TCE very readily, as well as some of the other chlorinated solvents. So we needed a field system that would allow us to inject isobutane gas, methane gas, and oxygen, as well as two bioaugmentation cultures, 493 and 494M, and some inorganic nutrients. The other thing that we were interested in the field was trying to figure out a way that we could rapidly grow up some of these bioaugmentation cultures, so increase the population in the field. And we tried to do this by the addition of isobutanol, and I'll explain that a little bit on the next slide, how that actually worked. So one of the things that we know is that when you grow organisms up on isobutane, there's an enzyme induced that's called short-chain alkane monooxygenase, or SCAM, and this is based on some of Mike Hyman's work at NC State. Now, when that SCAM enzyme is induced, it degrades isobutane, and the organisms will start to degrade 1,1-DCE, but you form this toxic epoxide. So this is a bit of a challenge because if you don't have a high enough population, you kill off a variety of your cells. So one way to try to get around this in the lab or in the field is you can grow these same organisms on isobutanol. So that's the first step in this pathway. So the SCAM enzyme converts isobutane to isobutanol. But if you grow the organisms on the isobutanol itself, then this enzyme is not induced. So you can grow up the population. The SCAM enzyme is not induced, but the cells will still grow because they can take isobutanol through central metabolism and grow. And you're not going to see degradation of 1,1-DCE. So the concept here is basically to go into the field, bioaugment this culture, ENV 493, in a state where its enzyme is not induced, circulate isobutanol throughout the treatment plot to try and grow the culture to a high level, and then add isobutane. And ideally, the population at that point will be high enough to degrade all of the 1,1-DCE and then start degrading all of the other contaminants of concern. So by doing this, the concept is to overcome this, this toxicity that you see with 1,1-DCE. So the system consists of two main components, a groundwater recirculation component. This consists of an extraction well and 5 injection wells. And this system is used to add basically our cometabolic gases, that's isobutane and methane, to the flowing groundwater. It's also where we can bioaugment the two cultures, ENV 493 and ENV 494M, as well as add inorganic nutrients in the form of diammonium phosphate, and isobutanol, which we discussed on the last slide. It's also important to note that this system has a component where you can add hydrogen peroxide. And this is basically to prevent biofouling of particularly the 5 injection wells during operation. The other part of this system is a biosparge system. And this is basically consists of 5 biosparge wells, and these are used to add oxygen to the plot.
So here's an overhead view of the demonstration well layout. It includes a central extraction well, which runs at about 12.5 gallons per minute during operation. That removes water from the aquifer to the Connex box, where it's amended with all of the different amendments that need to go into the aquifer with the exception of oxygen. Those materials are then injected through a series of 5 injection wells. The system then includes 5 biosparging wells, and these are used specifically to add oxygen to the aquifer. When sparging, you can get a higher mass of oxygen in than you can if you're trying to dissolve it in the groundwater ahead of time in the recirc system, which is why this design was selected. The system includes a series of monitoring wells that are nested within the demonstration area. It includes some dedicated DO probes so that we can monitor dissolved oxygen within the treatment plot. There's also both up gradient and down gradient monitoring wells. The up gradient well lets us know the concentrations that are sort of coming into the treatment area, and then the down gradient well lets us know what kind of declines you see just down gradient of the system. Now, so basically, if we're adding amendments, you see that you're going to add to those 5 injection wells pretty much equally. So the extracted water from the extracted well will be amended and then pumped pretty much equally into the 5 wells, where we'll get some radius of influence. Of course, we have a draw toward the extraction well because we're pumping the water there. And then the oxygen gets sparged. So hopefully then we get sort of a mixing of these types of materials so that we get optimal conditions for treatment of our contaminants of concern within this plot.
So one of the neat aspects of this system is it's completely off the grid. So it's run by a solar panel array, which we see in the upper right. So 16 solar panels. Those panels charge batteries, and those batteries then basically operate the entire system. So they operate the pumps, they operate the solenoids that turn on the gas sparging, and you can log in remotely and change some of the different functions of the system, which is really nice. So in the next series of slides, we've taken some video when this system was running, and we're going to show you actually what the system design looks like, what the gas addition components look like, and kind of what's going on the inside of the box.
As mentioned previously, this system is designed to inject 2 different cometabolic gases in order to stimulate bacteria to degrade chlorinated solvents and 1,4-dioxane. So the first of these is isobutane. Now this is a liquefied gas. Within this cage there are 4 different cylinders that are manifolded together, and the gas from this cage goes in through the control system and out to the 5 injection wells. So this gas stimulates organisms that degrade many of the chlorinated solvents, as well as 1,4-dioxane, but the organisms that grow on isobutane are not particularly effective at degrading trichloroethylene, or TCE. Therefore, we have a second gas that's going into the system as well. This is methane gas. So this cage contains 12 cylinders of methane gas that are again manifolded together. They move through the system and they get injected with the injected groundwater into the aquifer. And this stimulates methanotrophs that are known to degrade trichloroethylene and also some of the other chlorinated solvents as well, but not 1,4-dioxane. Thanks. So we've moved to the other side of the Connex box. So we have the alkane gases caged on the far side of the box. We're now on the front side of the box, where we have a series of oxygen cylinders manifolded together. So the cometabolic process that we're stimulating is aerobic, so we need to make sure there's enough oxygen in this aquifer for bacteria to grow upon. And so we move this oxygen in through the system, and there's a series of 5 different sparge wells, so the oxygen gets sparged into the ground creates aerobic conditions, and then these methanotrophs and organisms that degrade isobutane will grow aerobically and biodegrade our chlorinated solvents, and for the isobutane degraders, the 1,4-dioxane.
We're now inside the Connex box. We're in the larger portion of the treatment system. This is a intrinsically safe environment, so all the equipment within here is explosion proof. Should we need it. Originally, this system was designed for pure biosparging, and it was used on a previous ESTCP projects, and we did some modifications to the system where we also are able to recirculate groundwater and add amendments to the recirculated groundwater. So the original piece of the system is here on this wall. It consists of a series of mass flow meters and solenoid valves that control flow to up to 22 wells. On this project, we're only adding the oxygen to 5 of the wells. So you can see those at the bottom here. And the solenoid valves that control the flow to each of these individual wells. The oxygen comes from this side of the Connex box across the ceiling, goes through a flashback arrestor, which is a safety feature. Should there be any flames or explosions anywhere in the system, it will not let it get back to the oxygen cylinders. Another solenoid valve, and really the heart of this system is the mass flow controller. So this communicates with our PLC. We have put a set point as to what the flow should be at the well, and that controls the flow, and the solenoid valves control which well the oxygen goes through. So that flows through here, through another series of flashback arrestors, down here to the solenoid valves that send it out to the 5 biosparging wells within our well fit. So that was the biosparging portion of the hybrid system. The rest of the system is the groundwater recirculation and amendment delivery system. So the groundwater from our extraction well comes in along here, and it comes up through here where we have a dissolved oxygen probe where we can continuously monitor dissolved oxygen concentrations through our PLC and SCADA system. Then it goes through here through a system of filters. We've got 20 micron and 5 micron filters to filter out any particulates or biomass before it goes through the remainder of the system. This is a flow meter. Our flow set is right now is 12.5 GPM. This is a flow controller, so this communicates with our PLC. At our set point, this valve controls the flow to 12.5 GPM. Go a little further down the system, and you've got this static mixer where our 2 gases our methane and our isobutane are added. These are controlled by 2 mass flow meters up here. Again, this communicates with our PLC. We can make our set points, our duration, our frequency of the addition of these gases. These are controlled by solenoid valves, 2 inside and 2 outside, so we have a redundancy for safety. And we add these gases as needed into the recirculated groundwater. It goes further downstream, and this is where it splits into the 5 injection wells. Again, we've got flow meters and flow control valves on each of these. So these are currently taking 2.5 GPM to each of the injection wells. Throughout the system, we have pressure transmitters, so we can see what the pressures are. We can see if pressures are backing up in our filters. We can see pressures are increasing in our wells for any reason. All this information is logged and recorded by our SCADA and PLC system.
Yeah.
So besides oxygen and alkane gases, we also have to add inorganic nutrients. In this case, we're adding diammonium phosphate. These nutrients are mixed up in a chemical feed tank. This is a 150-gallon tank. We have intrinsically safe peristaltic pump that delivers it to each of the 5 injection wells. So this is where it's delivered. We have each of these solenoid valves controls delivery individually to the 5 injection wells. Again, the flow rate is set on the peristaltic pump, and we add these about once a week. Biofouling control is important in any of these systems, and the way we do that is through the addition of hydrogen peroxide. This system operates approximately 12 to 13 hours a day. Because it's run off of solar, we don't run it all night long. So at the end of each cycle, we add 600 ppm of hydrogen peroxide to the system for approximately 20 minutes. So this will be added again through a peristaltic pump. It's added to the beginning of the system where it runs through all the filters, all the piping, and into each of the 5 injection wells, enough to fill each of those wells and the borehole volume around it. And then we stop pumping for the next 12 hours. And that inhibits the biological growth throughout the system as well as in the injection well.
1, go.
So this is the control room, or the heart of the system. You've got solar panel charge controllers that connect to the 16 panels, and we have 2 different systems. We have a 48 volt system and a 24 volt system. The 48 volt system runs the pump and the HVAC unit. The 24 volt system runs everything else, all the controls, the computer, all the flow meters in the other room. We've got a set of 12 volt and 48 volt batteries in here. The 12 volt batteries are wired together in parallel and series so that you can get 24 volts for that system. And then we have 2 PLC panels. This panel controls basically all, and the panel to your right controls the pump and all the 48 volt system components. That's all connected to a laptop computer. This has a SCADA system on it, which allows us to log and record all the data, and to see this data remotely. We are able to dial into this computer, see how the system is operating, make changes, shut down the system if we need to. Several safety features built into the system that it will shut down automatically should certain higher low level conditions occur.
Great.
So now you've seen some videos on the design and operation of this hybrid cometabolic system. I'm just going to finish up by showing you a little bit of field data. And basically, here's the series of events that occurred. We started out with baseline sampling, then did a phase for about 7 weeks where we did groundwater recirculation and just oxygen biosparging, in part just to look at equilibration of the different contaminants within the plot, and then also to see how well we could distribute DO throughout the system. We then, in phase 2, bioaugmented with ENV 493 on 3 separate occasions, and we injected 500 liters of culture that was grown up in our lab here in Lawrenceville, New Jersey, along with isobutanol, followed by isobutane. We did a one-time bioaugmentation with ENV 494M, again a 500-liter volume of that culture, and then we did about 2.5 months of post-treatment monitoring. Okay, so I'm just going to show you a little bit of data, and basically I'm going to show you the upgradient well, which is the black line, and then just the extraction well, which is kind of a mirror of what was going on throughout the treatment plot. Obviously, there's a lot more data than what I'm presenting here. However, if we look at these operational phases, again, we did baseline sampling, and then at day 0, we started our recirculation with just oxygen addition. And then basically we did our bioaugmentation with 493 on 3 separate occasions, as you see here in red. And on 2 of those occasions, after we added 493, and now the organism was grown on isobutanol, as I mentioned, we added isobutanol to the plot to try and get the organism to grow up before we started isobutane addition. And you can kind of see in this second red line is where the isobutane addition started. And then we inoculated with ENV 494M here around day 100, so the methanotroph. And basically, if we look at the data for 11-DCE, we see a very nice decline after we first bioaugment. So going, here we were at almost 1,200 micrograms per liter as the plot became well mixed. And after we bioaugment, we see that we've declined down to, a couple hundred micrograms per liter. But we then start to see a little bit of rebound here between day 100 and 200. We bioaugment a second time. And once again, we see a very, very good decline. But everything stabilized, at 150 to 200 ppb. And even after a third bioaugmentation, we couldn't get the DCE concentrations to go any lower. Overall, we saw about an 86% decrease in 11-DCE. However, I think what happened was, as a function of time, we were probably pulling more 11-DCE into the plot. There was also probably some back diffusion from clays, and and other materials, some rate limited diffusion from sediments. So we always had kind of this background of 1,1-DCE, which despite all of the bioaugmentation efforts, we couldn't quite get down to a low enough concentration. Now for TCE, we see these same operational phases. Basically, we do our first bioaugmentation, we get some decline. We then bioaugment with ENV494M, which was, the culture that we knew degraded TCE very readily, and we had a really nice drop for a short period of time. But much like 1,1-DCE, we then saw some rebound and then kind of a stabilization of concentrations. Now, a lot of TCE was degraded. Again, at the down gradient well, we had about a 72% decrease, and throughout the plot wells, it ranged from like 22 to 85%. But we didn't achieve those low part per billion concentrations that we were desiring in the plot. Now, finally, for 1,4-dioxane, over the course of treatment, we saw some fluctuations up and down, but honestly, there was not a lot of treatment of 1,4-dioxane throughout the course of this demonstration. So the take-home lesson from this demonstration, first of all, the system worked beautifully. We were able to get very good distribution of gases. I didn't show you a lot of those data just for a function of time, and I'd encourage you to look at the project website. But we got very good distribution of gases. We seemed to get culture distribution. We had periods with very good biodegradation of our contaminants, but we weren't able to overcome this issue with 1,1-DCE. So I believe if in that first bioaugmentation we could have decreased 1,1-DCE concentrations down into the low ppb range, we would have seen a very good reduction in all of the other contaminants, because that's what we saw in all of our laboratory testing. But we always had this background of 1,1-DCE, which was likely killing the bioaugmentation culture and any native organisms that likely would have come up to degrade the other contaminants. So the take-home lesson is, if you get to these types of sites, these systems are great for dilute concentrations. And this system, as I said, operated beautifully throughout the entire course of the demonstration for what it was intended to do. But the microbiology didn't work that well. And I think most of that had to do, again, with this 1,1-DCE toxicity. So let that be a lesson learned, that if you approach a site that has very high concentrations of 1,1-DCE, you might think of another approach, perhaps doing anaerobic treatment first to try and reduce 1,1-DCE and other solvents, and then aerobically going after your contaminants such as 1,4-dioxane. So I hope what I've shown you today is that co-metabolism can be an effective approach for the treatment of a whole variety of different contaminants in groundwater. These include 1,4-D and TCP, NDMA, EDB, and dilute CVOCs. And that the technology is very, very well suited for dilute plumes. And you can achieve nanogram per liter concentrations at times. So there's a lot of positives to this approach. The last example I showed you was one where there's a bit of a lesson learned, and that is that sometimes if chemical concentrations are very high, and particularly if you have high 1,1-dichloroethene, this technology may not work terribly well, and that's because the 1,1-DCE causes microbial toxicity. And as you could see, that can be very, very hard to overcome in the field. So in that case, even if your system works well and you get your desired gas concentrations, you may not see the desired results in terms of reductions in 1,4-dioxane or the other contaminants that you're trying to treat. The other thing I didn't talk about very much today, but that's pretty important, is that the impacts of this technology on groundwater geochemistry, things like metals mobilization or sulfide generation, are a lot less than anaerobic treatment. This approach has really progressed a lot over the last 2 or 3 decades, and it's really widely applicable now for treatment of a variety of dilute plumes, which tend to be a major issue these days. So I'd like to finish up today by acknowledging the ESTCP program, both for funding some of the demonstrations that I presented today, as well as for funding this presentation through a technology transfer grant. In particular, I'd like to acknowledge Dr. Andrea Leeson, the director of the CERTUP ESTCP Environmental Restoration Program. I'd also like to thank both Dave Lippincott and Greg Lavornian, Aptum. They work in my group, and you saw Dave in some of the video clips. Both he and Greg are instrumental in the design and operation of these systems and really do an impressive job. I'd like to thank Michael Pound and Tony Danko with the Navy, Rose Forbes and Adria Bodur with AFCEC. AFCEC funded also 2 of the demonstrations that I talked about today. And then colleague Jim Begley with MT Environmental Restoration. So I'd like to thank you for listening in today. Hopefully some of the information presented was thought-provoking. Hopefully you consider co-metabolism for your next remediation project if that's your line of work. Included here are some peer-reviewed literature, some papers, as well as a couple of reports on the CertUp ESTCP website that relate to projects that I discussed. I would also like to encourage you to reach out to me if you have questions concerning co-metabolism. We can have a discussion, and if I don't know the answer, I can probably find someone for you who does. And my contact information is in the upper right hand of the slide. So thank you very much. Have a great day.